All right, we're going to talk about complex inheritance of patterns, and this is going to be when things are a little not quite as simple as just dominant or recessive. So first up is incomplete dominance. And with incomplete dominance, you'll get a blending effect. So if you cross red and white snapdragons, the first generation is all pink. Remember, your heterozygotes show an intermediate phenotype. But instead of it just continuing to blend, if you cross two pink together, your white and your red still show up. Your heterozygous will always be somewhere in the middle. Codominance works the same way, except instead of a blending, they're going to show equally. So you'll have red and white flowers. Multiple alleles work with blood types. So genes are going to exist in more than two forms. With blood types, we can have IA, which is for A type blood, IB for type B, or you can have a little I. And these are going to code for these proteins on the surface of blood cells. Now notice, sometimes you don't have any. That is where this guy comes in. So the problem we always do is if you have a heterozygous male or female for type A by heterozygous opposite sex of type B, what do you come up with? And you have to make sure you're looking at this as one allele, IA, little I, right? That's what that parent can give. Or IB, little I. I'm going to pause this so it's like magic. Ta-da! So what you can end up with, it is possible for a heterozygous A individual and a heterozygous B individual to come up with, there's an equal chance for type AB, for type B, for type A, and for type O. Pleiotropy is when you have one gene that has lots of phenotypic effects. Your example here is sickle cell over here with your when you just have that one amino acid wrong, it changes the shape of your red blood cells, which causes you not to carry oxygen as well. It can block up your blood vessels. You have lots of effects there. Cystic fibrosis is another genetic disorder where one gene is going to cause a thickening in all your mucus linings, and that can make you not absorb your nutrients as well. Lots and lots of stuff, which we'll talk about when we get to our genetic disorders. Sometimes you have two or more genes controlling one trait. So with epistasis, you have one gene at one location controlling whether another gene is expressed or not most of the time. So if we look at these mice, you You have a gene for color, the C's here. So if you have at least one big C, you're going to be either brown or black. But if you are recessive for both, you get white mice, no matter what else is going on there. If you have that one big C, then your other alleles, your brown or black, can show up and be expressed. The same kind of thing happens in labs. If they are recessive for this one little E gene, then they're going to be a yellow lab no matter what. But if they are dominant for that, then the chocolate or black color can show up. Polygenic inheritance. Poly means many. Let's see here. Hang on just one second. Okay. So you have many genes controlling one trait. And the main example here is usually going to be skin color and height in humans. This is how you can have two individuals of, if you have two individuals that are heterozygous for both, or for all of the traits involved, like if you're heterozygous for all, let's say 5 traits that control skin color or height, you would be an intermediate there. But you could have children, or you have a chance of children who are very light or very short. Most of your children should be in the middle. And you also have that chance, those extreme chances of very light and very dark, or very short and very tall. Hang on. This chart here shows something called a normal curve, or a bell-shaped curve, and that is always an indicator of polygenic inheritance, that trait being controlled by more than one gene. All right, that's it for this one. See you tomorrow.